Okay, so hello everyone. I hope you. So my name is Denis Bouman. I will just give an introduction on this webinar. So hopefully you see my slides. Just for those who do not know what is cost action, it's about it's essentially about networking activities. We are in Enota a cost action, and we have essentially for funding not for research as such, but essentially for networking activities. So it's a way to work, to build a network. And what is very important is the people who are in cost action, they want to be in contact with each other. So this is the principle, and this is the philosophy of cost association. So the next is here. Yeah. So the challenge of Enota is it's all about precision or personalized medicine for patients with inflammatory diseases using therapeutic antibodies. We are organized in 5 working groups. The working group that will be the highlighted today is a working group 2 because it's on the pharmacometrics. So Erwin and Elizabeth have the idea of organizing this webinar series, and it will be essentially on the proper dose at the proper time for the patient who receive therapeutic antibody. You see the other working group here. So what can be funded by cost if it does not provide money for consumable person? It's essentially for networking activities. We can have event meetings, also the training school. And I would like to point out what is very important to me is individual mobility for researchers. And we have what we call STSM, short-term scientific mission, that enable people from one laboratory, one institution in one country to have a mobility for, let's say, 3 weeks, 3 months on average, and to work on with another laboratory institution. So this is a way to trigger, to foster collaboration, and this is the spirit of association. So we have our website. You can have all the details, all the terms to apply for this STSM, and there are also other grants that you can apply. And then I would like to say that we are entering now the 3rd grant period. So you know that cost action are for 4 years. So we have almost achieved. We are halfway, and we are entering in November, the third grant period. So there will be one management committee meeting and working group meeting in December 11 and 13 in Paphos, Cyprus. There will be a training school on pharmacometrics. So the venue is also under discussion, but there definitely will be an important event of this 3rd grant period. We will release the STSM short-term scientific mission call in November so that people can apply, and just remind you that the mission has to be completed by the end of September. This is very important for budget constraint. And the second webinar series will start because Elizabeth and Erwin done their work, and after that there will be another one that will be organized by Pavel Kamalek. So it will be a different topic, and also for the benefit of the people of Enota. That was my last slide. I think I can give you the floor, Erwin, or you want me to present the first speaker?
It's okay, I can do it, no worries. So thank you for the nice introduction, also from my side. Welcome everyone to our new webinar, the first after summer. I'm very happy to see you all back. So this webinar for September is focusing on the regulatory and the industry perspective of TDM, or model-informed precision dosing, specifically for biologicals and biosimilar drugs in Europe. And we're very lucky to have two excellent speakers on these topics. And we will first start with Paulo Paixão, who is also a member of INOTA. So Paulo is an assistant professor in pharmacokinetics and biopharmaceutics at the pharmacy department of Lisbon in Portugal. And he is also a clinical pharmacology assessor at INFIMED, which is the Portuguese regulatory agency. Also is Paulo a member of the methodology or methodological working party of the European Medicines Agency. So from all of his regulatory work, he of course has a lot of very relevant expertise, and we're very happy that he wants to share all of that with us today. And moreover, he's also doing research at the university in Lisbon, where the topic is, of course, also pharmacokinetics and therapeutic drug monitoring. With that, I want to leave the floor to Paulo.
Thank you, Erwin, for your kind presentation. I will share my slides. I hope that everybody's seeing them. Since fortunately I'm not in my office but in an hotel room, and the network is not that good, I will ask your permission to turn on the camera in order to go swiftly without issues with the connection, if you don't mind. So let me just try to do it. Okay. So the purpose of my topic of my presentation today is to talk a little bit about the regulatory approval of biologics and biosimilars, and how can this somehow influence the perspective on TDM and MIPD for the public. As a disclaimer, because of my regulatory work, and I must say that this is just my personal views, and if I say anything wrong, that cannot be attributed to the INFIMED or the EMA. Is a usual thing that we need to do on these situations. So are drugs approved in the European context? So we started the European Medicines Agency by an article in the Council Regulation in 1903. And so in this article, the European Agency for the Evaluation of Medicinal Products was established. Before that, every country had its own national agencies with more or less relevance, and was at the time responsible for the approval of drugs in each of any of the European countries themselves. But with the EMA, we started to have a global way of dealing with new drugs and new drug applications. So at the time, we have 27 European member states. We were more, but as you know, with the Brexit, we lost UK in 2018. But the EMA also include these that are called they are from the European Economic Area, which are the Iceland, Liechtenstein, and Norway. So in the total, talking about 30 countries that join together and take decisions together whenever they approve new drugs. What are the various roles of the EMA? So the agency is responsible for evaluating the marketing authorizations for human and veterinary applications. It also coordinates the European pharmacovigilance system, is also providing scientific advice for the development of medicines, is also evaluating the application for orphan designation in EU. So orphan drugs are drugs where due to the fact that the disease that they are target to is rare. These orphan drugs often have advantage in terms of prioritization and simplification of some procedures for them approval. Is also responsible for the evaluation of the pediatric investigation plans or waivers in case that the drug is not to be used in pediatric patients, and is also responsible for the evaluation of the arbitration and referral procedures. So whenever countries are not in agreement between themselves. It's responsible also to provision of good quality and independent information for medicines that it evaluates in order to give this information for patients and healthcare professionals. Finally, the coordination of inspections for the clinical sites and the analytical sites is also made by the EMA. These have changed over time, and here we have a timeline. It all started in 1995, where EMA was physically introduced in the UK and started to work. At the time, it was called EMEA, but the idea was already there. And it started with centralized and mutual recognition procedures. This means that, of course, it started from a change from national directed approval of drugs to international directed approval of drugs. So this centralized is already an idea of approval of drugs using this centralized system. But then also there was the need for recognition of procedures that were already made at national levels. This was the starting point for the assessment of medicines. Some important points that we have here is, for example, the appearance of the decentralized procedures that are mainly used for generics, and is a way of within the realm of the EMA, but taking each national country in his work approve a drug based on an application for that national country that then is expanded for other countries within EMA without the need for going for a centralized procedure. There are here also some main points, like the first two biosimilars in 2006. These first two biosimilars were the first in worldwide applications, and the first one was a biosimilar for Omnitrope. The first centralized authorized generic, because typically generics were approved at the national level or by mutual recognition procedures or decentralized procedures. I will talk a little bit later about that. But at some point it also started to be approved centrally. Then the first gene therapy medicine in 2012, the first stem cell medicine in 2014. The important thing, the proactive publication of clinical data that started in 2016, which is an important way of getting information from the EMA. And the last one, 2020, the relocation to the Netherlands that was completed and is now in full speed. So the EMA network is consisting of all these committees. Some are related to veterinary drug products. Most of the others are related for human products. This one, the Committee for Human Medicine Products, is the one that is responsible for the approval of new drugs and drugs at the centralized level. This decision made scientifically here is passed to the management board that then transfer it to the Commission Parliament that finally approve the drug entering in the market. So the final approval is in fact politically and made at the Parliament of the European Parliament. Regarding the type of procedures, I already mentioned the centralized procedure. This one means that it's received not in individual countries, but at the EMA itself, and then the EMA distribute to what we call a rapporteur and a co-rapporteur that are from 2 different European countries. These are responsible for making the reports and giving the first assessment of the product, but all the remaining countries at EMA can then participate later and participate also in the final decision of the approval of the drug. There's this mutual recognition procedure. This means that the drug was already approved at the national level in one country, and then the company wants to apply to a different country, and so it just consists of a passage of the previous evaluation from this original national country to the other ones that can accept or not that assessment, and if they accept, the drug is able to go then to that second market. The centralized procedure is the one where a new drug application is made directly to a country that will be the rapporteur. Besides this rapporteur, all the other countries where that the company intends to introduce the drug in the market are also involved in the procedure, but as secondary members. So the rapporteur is responsible for doing the report, presented to the secondary members that can accept or not. And if they accept, then in the end, the drug is also introduced in their country. Finally, there is still the possibility of the national procedures going outside of the realm of the EMA, and in this case, the assessment and the market is a single country where the application was done. Based on these, are any specifics for biologics? Well, the centralized procedures is compulsory for several situations. For example, for human medicines containing new active substance to treat HIV or AIDS, cancer, diabetes, neurodegenerative diseases, autoimmune or other immune dysfunctions and viral diseases. So in this case, all new drug applications should go by a centralized procedure. It's also mandatory for medicines derived from biotechnological process such as genetic engineering, advanced therapy medicines, and orphan medicines. These are all medicines that needs to go from centralized procedures. So whenever we are talking about biologicals, because they come from most of the time biotechnology processes, we are talking about centralized procedures, and also for the biosimilar medicines, which for biologicals are also mostly coming from biotechnology processes. So these are usually also evaluated by centralized procedures. What type of applications exist in the EU? Well, we have these several articles that are from the directive of 2001/83/EC. The first one, Article 8.3, is a full application, which means that the company needs to present quality, preclinical and clinical data. So with all the data gathered and provided by them. But we have also other possibilities like generic medicines products, Article 10.1, where basically the companies need to show bioequivalence between the test and the reference product. We have the hybrid medicinal product, the Article 10.3. In this case is probably an already known molecule, but is now intended to be administered by a different route, for example, or using a different dose. And so it does not need to show everything again about the quality, the preclinical and the clinical data, but some particular parts of the clinical data, for example, and the quality that needs to be updated. We have also similar biological medicinal products. Well-established medicinal use. This is for molecules that are already for a long time in the market, more than 10 years. and often there is also a situation that the reference product is no longer available in the market, so the possibility for developing a generic medicine is no longer possible, and so this rely on published data and the bridging to that published data that can be done either by some bioequivalent study or some in vitro in vivo correlations, for example. We also have fixed combinations when we combine two drug products that are given together but in two different formulations. And we also have the informed consent whenever a company gives their procedure for a second company and allows that second company to put their initial product in the market by themselves. So how is the scientific information provided for review? We have this CDT triangle where the model 2 is responsible for the quality data, the model 3 the model 2 is responsible for the resume of the every data that is available in the procedure. The model 3 is that for the quality, the model 4 for the clinical study reports, and model 5 for the clinical study reports. And this is what is provided for in any application, and then the assessment is made based on relevant guidelines and scientific state of the art. So where can we see the guidelines? We have two main places where we can see. In the EMA page related to the scientific guidelines. Those are the ones that are produced directly by the EMA network, but there are also guidelines coming from the ICH, which are what we call the harmonized guidelines in the world that make part of the ICH. EMA makes part of the ICH, the FDA is also, PMDA and several other multiple regulatory agencies are here. And so these guidelines, when accepted, take over the ones that are produced by the EMA themselves or itself. So if we talk about biologics, I made here a small list of the most relevant guidelines that are available for them. The first one is a guideline for the clinical investigation of the pharmacokinetics of therapeutic proteins. The second one, the guideline on similar biological medicinal products. The third, the guideline on similar biological medicinal products containing biotechnology-derived proteins and active substances, focus on non-clinical and clinical issues. Then the guideline of similar biological medicine products containing monoclonal antibodies, non-clinical and clinical issues. And finally, a dose response information to support drug registration. The first ones are all from the EMA, the last one is from the ICH. And what does these guidelines provide? Well, they provide the main directions for the approval of drugs in this particular part. So in this one, we are focused on the pharmacokinetic characterization of therapeutic proteins, and it puts here some of the main points. I will not describe in depth this guideline, but what comes from here is that absorption is typically very simple because therapeutic proteins are usually given intravenous or at least subcutaneous, and so the description of the absorption process is all relevant if you give subcutaneous or and then the comparison with the intravenous part is just to give an idea of the bioavailability. Regarding this position, the main points are here. We have subpopulations here, but in fact, most of the cases, therapeutic proteins are usually not tested in renal and hepatic impairment subjects because it usually does not influence the disposition of these proteins. And the interaction studies are often very reduced in pharmacokinetic terms. And most of the times what we see is interaction studies due to pharmacodynamic interactions. One point that I want to point here is the PK/PD relationship, because in fact it is recommended that the relationship between drug concentration and pharmacodynamic responses should be evaluated in these cases. It is also requested that if feasible, markers for both efficacy and safety should be measured. And this is the difficult part that during drug development often is not made with great efficacy. Then there's this other guideline on dose response information. I need to pass fast on this. For example, important thing is the use of concentration response data for giving an idea of the PK/PD of the drug. This, although being requested, does not require that the product in the end be subject to therapeutic monitoring, but is a way of getting information for providing a direction for the doses to be used later. This is the guideline of similar medicinal products. It's a very simple, and it just says the bio approach is not the same as that one that was used for generic approach, because the biosimilar, the biological products are more complicated. The products or the medicinal product is not exactly the same between the test and the reference product, and so it needs to be assessed in a continuous process, going from the quality, passing for the non-quality, and then for the clinical part itself. For particularly monoclonal antibodies, there's also this guideline that makes this connection that you need to make comparisons from non-clinical studies before going for the clinical studies. In the clinical studies, the pharmacokinetics is a major one and a step one for this clinical comparison. You can have also a pharmacodynamics evaluation, but typically it's not enough, and then you really need to do a clinical efficacy study in order to prove the biosimilarity between the two drug products. How can we get scientific information? Well, we can get scientific information going for the European Medicines Agency. In the medicines webpage, you have access to all the relevant information made during the assessment of the procedures. And the clinical data, you can also find it on this European Medicines Agency clinical data page, where you may look for the original clinical data that was produced or provided in the applications, with some data being anonymized in order to protect the patients that were included in the studies. So some examples: Remicade, the first infliximab product that was introduced in the market. The application was authorized in 1999. The first public report was only made in 2005, so much later than the date of the approval. It combined several assessment reports that were made between the initial approval and that time of 2005. The data is not well described and is difficult to fully follow, and in fact represents the at-the-time approach for public assessment reports. So you have here the idea. They made or they provide a review of the clinical studies that were made, but the information on these clinical studies is very sparse and is not that much informative. It's basically informing if there was a patient or a healthy volunteer population included, the number of subjects, but nothing regarding doses and things like that. And the information that was provided in pharmacokinetics, pharmacodynamic terms was also very limited. They told us that multiple doses were studied, but some strange things were seen. For example, the TNF-alpha at baseline was initially reduced, but then was increased, which was not logical, and the reason was likely due to the formation of infliximab TNF alpha complexes that were not able to be correctly discriminated in the analytical study. In fact, the pharmacokinetics was also limited because of the analytical issues. And so based on that, the information provided was provided also with that in mind, that it could be influenced by that flawless analytical method. So this is something very new at the time, and the reason for accepting this was in fact the limitations on the analytical part, but the clinical part that came after really provided efficacy and safety data that allowed for the applications to be approved. Then this Remsima, that was one of the biosimilar medicinal products that were produced at the time. This one is from 2030, the first approval, and we can see here a better description of the two pivotal clinical studies. Here they are presented in a descriptive way. The first one was on patients with IS, and the second one with patients with arthritis rheumatoid. So these two pivotal studies were also followed by a third study that was a pilot one, just to optimize the conditions for the efficacy and safety study. Here are the results. So we start to have the results of the studies being presented, the plasma concentration profiles over time. A little bit strange here, but this is because of the timeline that is being cut. And of course, this shows us the part of the pharmacokinetic comparison, showing that in fact the biosimilar and the reference product were bioequivalent in terms of AUC and Cmax. At the time, only AUC-T was required. Nowadays, we also request IUC from infinity, from 0 to infinity, to also show bioequivalence. Another thing that we see here is regarding the bioequivalence of the profiles where subjects were with or without antibody positive responses. So if they add ADA in their description, the bioequivalence was also shown in subjects without ADAs and with subjects with ADAs regarding the comparison between the test and the reference formulation. So this was related to the safety issue. Since there was no relevant markers for therapeutic activities, there was some PD study and some PD parameters were collecting. And although showing great variability, similar decrease was seen in both treatments. But because none of these PD parameters was considered to be clinically relevant, this PD comparison was not sufficient. And in fact, the proof of equivalence was also supported by pivotal efficacy study based on a clinical endpoint that was the 20% improvement according to the American College of Rheumatology criteria response at week 30. So showing equivalence here allows then this to be a biosimilar product. You know that Remsima also later produced a subcutaneous formulation. And in this case, this was not a biosimilar application, it was in fact a hybrid application, because it was a new route of administration. And being not a full application, the clinical studies were including 2 single-dose studies in healthy subjects, 1 study in patients with rheumatoid arthritis, and 1 study in patients with Crohn's diseases and ulcerative colitis. The studies are here well described, but what I would like to show is these plots next. The first one is showing the difference in pharmacokinetics between the intravenous administration and the subcutaneous administration. The idea here for the company was trying to have similar IUCs for an 8-week interval. That was the interval of administration for the intravenous administration. And so they have here the multiple dose situation in intravenous administration and several increased doses of the subcutaneous administration, and they come to the conclusion that the 120 milligrams twice a week was able to produce a slightly higher AUC, but the one that they would intended to test it in the clinical trials. They also show that the C-throw at week 30 was much higher on the REMC subcutaneous administration than the intravenous administration, and being the C-throw a relevant pharmacodynamic parameter, this was also indicative that 120 milligrams every 2 weeks was possible to go to the clinical trials. And yes, the time is going up. Yeah, I have a couple of more slides. I will try to be fast.
Perfect.
The company developed this POP-PK model also connected with a PD model. In this case, with clinical scores, this CDA score and the Mayo score. So was also intending to have this clinical relevant PD markers. However, due to some clinical conditionants, the relative standard errors was very high in Bootstrap for the IC50, indicating that this parameter was poorly estimated. So this, together with the fact that the clinical data were from open label studies, and all subjects were treated with intravenous in the first 6 weeks, and not part of the risk was before randomization, limit the application of these models for the subcutaneous administration. In any way, confirmation was again based on clinical efficacy data, both on rheumatoid arthritis and Crohn's disease. And the population POC-PK also showed that with this dosing, the threshold for efficacy was obtained for all of the patients, even for the ones with a high weight. So conclusions: the drug approval for marketing applications in the EU follows strict scientific and regulatory criteria, and is a multi-team, multinational shared decision process. It is based on quality, non-quality, and clinical authorization of the drug. Biologicals share many common requirements, some with added, others with less complexity. Developing PK-PD relationships is a must-desired goal, but not always fully explored. In the end, it is mostly the clinical efficacy and safety data from confirmatory phase 3 studies that define the BR ratio, but this leaves a lot of room for later optimization in phase 4 of the drug's use, which is something that is also proposed by the ICH dose efficacy guideline. And thank you. This is the last slide. Sorry for taking a little bit more than I was expecting. Thank you.
Thank you, Paulo. So we can no longer wait and give the floor to the next speaker. So Gregor Schaffar, he's heading the clinical bioanalytic laboratory at Sandoz, and he has been focusing on the development of and validation of state-of-the-art pharmacokinetics and PD biomarkers, as well as immunogenicity assays. So we have time, but if we want to have some time slot for a few questions. So thank you for being with us and to join Enota for this webinar.
Yeah, thanks for the introduction. I try to be fast, and I hope it's not too analytical, but at least can give you some considerations how biosimilars developed, what kind of bioanalytical considerations need to be done for respective assays, and looking at potential impacts on therapeutic drug monitoring here as well. So starting, next slide, it works. Also for me, the disclaimer, I'm working for Sandoz, and the views I'm expressing here in that presentation are my own views. As just said, shortly we'll give you an introduction into biosimilar development. Paolo already mentioned main guidance is on that one. Focus then on the specific PK, drug concentration measurement strategy here, considerations for TDM, but also will include immunogenicity assessments for biosimilars, and again, ending with what this means regarding TDM assay potential, also looking at biosimilars. Starting here, again, just that you're aware that the development of biosimilars required really a paradigm shift in pharmaceutical development. So when you look at the innovative development of biologics, the major goal here is really to determine the clinical effect, looking at the benefit risk of the molecule. Quite a short part of analytical. It's a big part of looking on a clinical basis on the benefit risk part. The biosimilar development is upside down. We have a different pyramid where we really start with a very extensive analytical characterization of the molecules we develop for biosimilars. This is to establish the similarity and to demonstrate that across 4 main categories. That is what is always needed for biosimilars. That's the amino acid sequence of the molecule, of the biologic. The primary sequence is identical. The high order structure needs to be indistinguishable. Glycosylation or other variants should be there in identical structures, but in comparable amounts. Differences need to be not clinical relevant. And of course, the bioactivity, so the mode of action of the protein needs to be comparable. As already said by Paulo, we are talking not about small molecules or generic, it's biosimilar, so there are always differences, and therefore for biosimilar development, clinical studies are needed. This is done in 2 phases. Phase 1, which is primary focus on pharmacokinetics and pharmacodynamics, if respective parameters are available. And the second one is about the clinical safety and efficacy, which is done in a larger phase 3 trial and a sensitive indication. And that's then confirmed similarity and can the whole package of analytical and clinical biosimilarity approaches is then submitted to health authorities. The biosimilar concept from a clinical perspective always contains these clinical studies, and in a biosimilar setup, they are done in a head-to-head comparison of the proposed biosimilar and the respective reference product. So we have trials where we have 2 arms of treatment, one with a biosimilar, one with a product, doing the sampling in the same way, and then comparing the products, which is also driving then the analytical strategy, to which I'm coming now. So what does it mean from a drug concentration measurement perspective? And just as a starter, the analytical methods we need for clinical biosimilar programs are really essential because they are measuring very sensitive parameters which we need to demonstrate finally the biosimilarity. It's a concentration measurement with PK, drug concentration assays, but also the immunogenicity when we deal with more immunogenic products. So we really need a very specific development and validation of those assays with accurate performance and during the validation, but also, of course, during the conduct of the study. These biosimilar analytical assays are not different to what you know from new biologic assay development, but we need specific adaptations for them, coming to that later. But just again, to show you the difference, developing a new biologic entity in NBE, the PK objective would be always to characterize the PK for the new entity, for the investigational drug. And the assay should be sensitive and specific to really detect this specific drug. Different for biosimilar development, we evaluate PK similarity. So it's not about just the PK profile, but it should be always the similarity between at least 2 products, the biosimilar and the reference product. And accordingly, the objective of the assays should be, of course, being again sensitive and specific, but to detect both the reference product and the biosimilar in a similar way. This can be done in different ways, and there was a long time a debate about how the best approach would be, because having more than just one drug in those trials means you have also the opportunity to work in assays with different drugs for standard calibrators and QC samples, which could result you're either working with one assay detecting both reference and biosimilar, or more than one assay, so the 2 PK assay approach, specifically for each drug, biosimilar or reference. The 1 PK assay approach has the advantage you have less variability due to the assay, and you can do the study analysis really in a blinded analysis. And this is 1 PK approach is actually what is also proposed by health authorities, by FDA and by EMA, and it accepted and is done and performed by all companies in the industry here, really using 1 assay, always in drug development of biosimilars to detect drug concentrations of biosimilar and reference product. The 1 PK assay approach means we are using the same methodology and the same procedure, always the same reagents, and we are only using 1 reference as standard and QC for the preparation. Most often it's done with a biosimilar, but you have also companies doing it with the originator. There are no specific guidelines from health authority how to validate that. Really, this 1 assay approach is able to detect comparable biosimilar and reference products. The most common approach in industry, and it's also accepted by health authorities, is really to do a very extensive validation that you compare, as shown here in the graph, for example, that you show when you're using different standards and different QC samples from the different drugs and do overlays that the standard curves overlay, or you do back calculation of QC concentration of spike samples, that the ratios are comparable. And having proven that, you can go with the 1 assay approach and doing the analysis respectively in the assays. What does this mean for TDM assays, which you're using? So especially in the field of autoimmune conditions, as IBD, psoriasis, RA, right? This is an important tool to support therapeutic decisions. And it involves here also not only the drug concentration measurements, but also detection of potential anti-drug antibodies. Per definition, and following the guidances, a biosimilar is a biologic that is highly similar and has no clinical meaningful differences in terms of safety, which includes also immunogenicity, purity, and potency to its reference product. And as just shown, right, by the analytical characterization of the product, but also by the clinical evaluation using this 1 assay, detecting both in the same way, biosimilar and reference product, we can guarantee that it's really comparable to the reference product. In that respect, we do not expect any differences between the biosimilar and the reference product using TDM assays, right? And this is also the question, do we need a biosimilar-specific cross-validated TDM assay? I mean, the question, of course, which can come up. There are different therapeutic drug monitoring assays available. I think most often it's still the enzyme-linked immunoassay, which is also the basis in most cases for drug development in general. So the same principle is used here, even though it's maybe by a TDM manufacturer, a different kind of ELISA approach is used here, right? The principle are always the same. And therefore, also looking at the data which are produced during development, we can expect that the biosimilars as a reference product are highly similar, and that no specific TDM assay is actually needed for the biosimilar. So you can use the 1 TDM assay, which is detecting the reference, but also maybe different biosimilar products. This is also shown when you look at different manufacturers in publications when they are doing that always you see a good correlation actually between references and different biosimilars on the market. So there's quite a confidence that there's no difference and no difference expected also by the way how biosimilars are developed. To even make sure that this will increase and to standardize and harmonize drug monitoring assays, right? I think it's important to have in the future more and more of these international standards. One in publication here from that, but for adalimumab is also showing that. And this is really helpful, I think, then to evaluate also the variability. So we do not expect variability from the different drug products, whether it's a reference or biosimilar treated patient. We see more variability from the assays, but international standards will help you also to standardize the variability, which is actually quite good. You see here an example of different assay used. There is always inter-assay variability and inter-lab variability. This is quite normal in a biologic assay, right? So I'm not concerned about that, but it will help actually to standardize TDM. approaches here as well. Leading me to the second part, which is a little bit focusing on immunogenicity. I think you're all aware of the potential immunogenicity of biologics. So it's nothing specific about biosimilars. I think it's just something which comes from the biologic itself. But it's of course also during the development of a biosimilar an important thing, and where we are focusing here is on the development of potential anti-drug antibodies against the biosimilar product. Again, advantage with biosimilar development is that we have a good knowledge and understanding of immunogenicity already from the reference product, which helps us to develop the biosimilar into that. Immunogenicity is a big part of biosimilar development, an important part. It's always considered when we have our head-to-head comparison clinical trials. The difference between NBEs is that, again, it's not looking de novo at immunogenicity of the product, because from our analytical characterization, we know that these are similar products. The immunogenicity assessment of the biosimilar will always focus on confirming that there are no clinically meaningful differences between the reference product and the biosimilar. So that's the purpose of this approach, but part of every biosimilar clinical trial, and some data are submitted actually to health authorities as well. As in other developments, I think the approach of bioanalytical assessment of immunogenicity requires a quite sensitive approach with a tiered approach with different assays, starting with a screening, a confirmatory assay, semi-quantification assays, and even more characterization assays, whether these are neutralizing or non-neutralizing antibodies. So quite extensive, more extensive than what you have with just positive, negative in TDM assays. But what I think is important, which relates again to therapeutic drug monitoring, is that when you develop a biosimilar, you always have the problem that we need to talk about relative immunogenicity. So immunogenicity cannot be standardized currently from an assay perspective, because you're using different formats in different companies, different operating conditions, different reagents, and they are differently calibrated. And therefore, when we as a biosimilar developer compare the data which we have with historical data, there could be differences. And therefore, we need the head-to-head comparison of that trial really to compare immunogenicity. And therefore, we always talk about relative clinical immunogenicity. We report not only just anti-drug antibody, but also the specificity, whether it's the frequency of neutralizing, but also time courses, for example. Important, and I think that's also important for looking on from TDM perspective on immunogenicity, is that needs to be always in the clinical context. Immunogenicity cannot be in an biosimilar exercise, cannot be looked at a standalone parameter. We always need to look at PK/PD, safety and efficacy, where we see in correlation or not. And this is, I think, something also different compared to an NBE development where the focus is quite different. We have less studies based in the biosimilar development. Therefore, it's so important really to look at that from a holistic perspective. Also here, of course, as with PK assays, you would have the opportunity to develop one assay or two assays to differentiate between the biosimilar and the reference product and the anti-drug antibodies against them. But also here, the requirements from health authorities are in the direction of one assay. This is also what industry is doing here. It's normally, again, an ELISA-based assay, most often a bridging assay where you just have a sandwich of your potential product. One assay is done, and again, we need special validations performed here with surrogate antibodies, for example, where we have depletion curves of in our assay, where we either spike with a reference product, the originator, or the biosimilar. If the depletion curves overlap, it's a demonstration of the assay that it's feasible to detect both anti-drug antibodies against reference, originator, or the biosimilar as well. This is how it's done actually to use that. In industry. Looking at immunogenicity of biosimilars approved, can skip that. I think here it's shown that we have here comparable immunogenicity in approved approved biosimilars, in that case infliximab, adalimumab. I think that's nothing new. For TDM, TDM and immunogenicity of potential anti-drug antibodies, which should be included. I think you're in a similar situation. It's much more difficult when you look at the available assays on the market, because what is most important here is really the sensitivity and the drug tolerance of the assay. Really, that makes a difference whether you are able to detect these kind of antibodies or not. And you always need to consider, as in a biosimilar development, from a holistic perspective, the clinical context. Immunogenicity data are not standalone data, and as proposed here by the publication from Brian Rowan, you need to look at, of course, also concentration and also clinical data you have maybe for efficacy to really evaluate what's going on in the patient using these assays. And maybe here I pause so that we still have some time for discussion.
Thank you, Dr. Schaffar. It's a very, very nice presentation. It's a nice overview for sharing all these insights. So I think we can start a discussion. Is there anyone who has questions for Paulo or for Gregor? Denny, I think you're saying something, but you're muted.
Oh, sorry. So please raise your hand if you want to ask a question. I have one question for Paulo. So if you want to have a kind of an approval, let's say for using a biomarker or using drug adjustment, is the company the only one who can apply to ask for or is it only the manufacturer or the company, the pharma company, who can ask for an extension of indication? Or
to be honest, I don't know. The logical thing would be coming from a company if it's an advantage for them.
okay,
but you can always present a new method for evaluation at the EMA, and then ask for their opinion on what the next step would be. Yeah.
Okay. Nick Olford has a question. Nick.
Thank you. I think you have a question for Gregor. I was interested in his in one slide where he showed differences between different assays trying to measure a standard concentration, for instance, 2 milligrams per liter. Couldn't really see any analysis of what numerically what the bias was. It looked. Can you give a number? Bias is the number I'd look for. What's the bias in different assays? If you have a standard assay, which you believe is the reference, what is the bias in the other assays? If it was a bias of plus or minus, of plus a milligram, when the reference is 2, that's a big bias. So it would be 50% bias. What's the bias of these across the different methods?
Yeah, I would need to look it up. I don't have it in my. I can share also the presentation or the publication where I have it from, so I can provide you that. Normally, the bias which is allowed at these biologic analytical assays is 20% plus minus. So, but I can check that and provide.
Yeah, that's the sort of assay or sort of bias you might allow within an assay. But when you're talking across, I'm thinking about trying to use concentrations when a patient may move from one clinic to another for one reason or another, measured in a different place. I mean, that's what's the most important number for me. Otherwise, I don't care at all. You do a great job measuring something, but if it's biased, I'm not interested in it, or I need to know how much it means in order to interpret it from a clinical perspective. That's my role. I'm a clinical person, tries to help people interpret concentrations. And if you don't know the bias, you can't tell me the bias, I don't know what to make of it.
Okay. No, I got it. I will check in the publication whether I can find something and will provide it afterwards.
Okay.
Can connect, sure. Yeah.
Okay, thank you all. So it's time to maybe to Erwin, can you close this webinar, and in between I will share my screen to announce the next webinar. Erwin.
Okay, great. So I won't take much more of your time. So thank you all for attending. Thank you again to the speakers for these. Next on, yes. Thank you again for these excellent talks. It was really interesting, and we really look forward to meeting you again in a month, because then we're in October, and then we will have our next webinar, which will be about software tools for model-informed precision dosing. So thank you all again for attending this webinar, and I wish you all a very nice rest of your day or evening.
Perfect. And have all of you an excellent new academic year, and see you next time. Bye-bye.
Thank you.